All right, everybody. This is the last section of the first week's material. This is going to be on microbial interactions. There's a lot of material in this one, so I'm hoping to get through it relatively quickly, but it might take a little bit longer than the other videos. All right, so by the end of this module, you should be able to compare and contrast parasitism, mutualism, and commensalism. Define the term normal microbiota, and discuss its establishment and roles, and describe how a biofilm forms, and discuss the healthcare implications of biofilms. All right, so we've seen this little image before. This is just to remind you that microbes may be friends or foes. There are a ton of microbes out there in the world, about 1 trillion species on Earth. The number of cells is about 10 to the 30 or 10 to the 31, 10 to the 30 microbial cells, about 10 to the 31 viral particles on Earth. And there's about 45 times more microbial biomass than animal biomass, right? So all these guys are microbes. They're dwarfed by plants, of course, and then we've got animals, which is a much smaller portion of the overall. And this is the same on our own bodies, or at least close to the same. So there's approximately the same number of microbial and human cells. It's about a 1-to-1 relationship. And one of the most recent examples that I could find, there are about 30 trillion cells in your body. And then there's about 38 trillion bacterial cells in your body. Now, from a biomass perspective, it's a little bit different. Your body mass is going to make up the large percentage of your total biomass. So here we have an example from like a 70 kilogram man, and about 0.2 kilograms or 200 grams comes from the microbes in your gut or in your body. So just to put that in perspective, there's about 1 iPhone worth of bacteria in your body. iPhone weighs about 187 grams. There's about 200 grams of bacteria inside your body. So normal microbiota is going to make up the majority of those microbes in your body. These are going to be the bacteria, the archaea, and different eukaryotic microbes that exist inside of you. They have their own lives that they live, right? But there are some things that they do that are beneficial to us. So we may think of them as like their function inside of our bodies, at least as they relate in a anthropocentric way. They train our immune system, so they're able to teach our immune system how to recognize friend and foe sometimes. Some of them can produce vitamins. They help break down complicated um, molecules that we're unable to digest without microbial assistance, and they can even impact our moods. There's this idea of a gut-brain axis that the bacteria in our guts can actually send neurotransmitter signals that interact with our brain that adjust the way that we feel. Now, there are a couple of different interactions in terms of like symbioses that exist here, and there's more than what I've listed, but these are 3 of the big ones, and they really kind of exist on a kind of a continuum anyway. So there's mutualism, which helps the host, and then there's commensalism, which is no perceived benefit or cost to the host, and then there's parasitism, which hurts the host. And so an example of a mutualism would be like E. coli, K12, or sorry, E. coli. It makes vitamin K in the gut. And so over here, we see plus for host, plus for microbes. So the E. coli has a place to live. It gets access to the food that goes into our intestines. And so that's how it's benefited. And then the host is benefited because it actually, the E. coli produce the vitamin K for us. Now, an example of commensalism is Staphylococcus epidermidis living on our skin. And so in this case, there's no real perceived help or downside to the host. For the microbes, they do benefit. They eat the glycerol that our skin produces. And so that gives them a carbon source. but we don't really see anything on our end. And then here we have pretty much all disease-causing organisms, or what we'd call parasites or parasitism. This is where there's a negative effect to the host, that's the disease state, and a positive effect to the microbe. In this case, we're talking about they're getting whatever benefit they get from causing that disease, usually some form of nutrients or carbon. Now, just to take a moment, in most cases, mutualism, commensalism, and parasitism live on a continuum. And so an example here is Staph epidermidis, which is a commensal organism. Generally, we don't think of it as a benefit. They're just eating the glycerol. However, maybe there is some host benefit. For example, the Staph epidermidis may competitively exclude pathogens from colonizing your skin. So maybe it's a little bit of a mutualism. However, if you get a cut in your skin, or if you are um, immunocompromised, and Staph epidermidis has the access to infecting you, it might actually move over into parasitism. And so the interesting thing about these symbioses is that they rarely stick within a single interaction type, but they exist within this continuum where they can move back and forth between different types of interactions depending on the context. All right. And so so moving on, we have the Human Microbiome Project, which aims to characterize all the microbes in the body. Um, Pretty much all parts of your body have microbes in them, and the Human Microbiome Project aims to characterize those. There's also an Earth Microbiome Project, which aims to characterize all organisms on the planet. Here's an example of the different organisms, different types of organisms that live within your body. Notice that in your intestines, we have our largest diversity with over 40,000 species. Now, when we're thinking about normal microbiota, we've got to kind of think what makes it normal. Um, so This often depends on location. So location, location, location. Something that's normal in your gut isn't necessarily normal for your mouth, right? And in fact, many gut bacteria that cause no disease when in your intestines, if they are ingested, will result in disease. So you don't necessarily want to put, microbes everywhere. Um, Another example is like some of our normal microbiota include things that are normally or that we think of as pathogens. So about 27% of adults asymptomatically carry Staphylococcus aureus on their skin. And so what that means is that their skin's colonized by Staph aureus, but they don't get any infection. However, as we talked about with the interactions, there are situations where that Staph aureus, like the Staph epidermidis, can actually become infective. Um, Now, one of the big things that our host microbes do, or our normal microbes do, is they crowd out potential pathogens. This is through a process known as competitive exclusion, and that's demonstrated in this image here. We have commensal gut bacteria, which are lining the lumen of the intestines, and up here we have this disease. This is a mouse disease-causing organism called Citrobacter rodentium, and it's actually unable to colonize this lumen because you have a healthy gut normal biota that's currently colonizing it. Um, Now, there are microbiome profiles that can determine or that can be related to diseases. For example, in people who have asthma or high allergies, they oftentimes have a lower diversity of microorganisms than people who don't have asthma or allergies. And so here we see kind of this graphical representation where healthy individuals have very diverse set of organisms. We see we see multiple different organisms and a bunch of them. And then down here, we see fewer of those organisms and fewer of those types of organisms. And these folks tend to, this microbiome tends to be related to like allergy. But it comes, to this question: is this cause or effect, or is this cause and effect, or is this simply a correlation? And so we need to continue studying this and try to see if we can determine a mechanistic relationship between these microbiomes and these, healthy or disease states. And if we are able to determine those relationships, we might be able to actually do microbiome therapy, where we adjust your microbiome to make you healthy. And in fact, we're starting to do that a little bit with what are known as fecal microbiome transplants, which we'll try to talk about a little bit more later in class. Now, establishment of your normal microbiota begins really pre-birth. There's microbiota that's found in the umbilical cord. The baby's first stool contains oral microbiota that would be found in the mother's oral cavity. And so, and then during birth, the baby passes through the vaginal canal and is actually going to be colonized with the mother's vaginal microbiota, which includes a lot of lactobacilli and lactococci and stuff like that, which are also found in the gut. So this is what I was saying. We get actual oral microbiota that begin to potentially colonize the baby pre-birth, right? So they're in that meconium, the first bowel movement the baby has. Here we have how different birth types can actually affect the diversity or the relative abundance of different organisms in your microbiota. On the left-hand side, we have CS, which is cesarean section. And then on the right-hand side, we have VD, which is vaginal delivery. Maybe not the best choice of, of acronyms for vaginal delivery, but what we do see here is we see this actual difference in the amount of these different phyla, right? So there's a lot more firmicutes in the vaginal delivery, and then there's a lot more of these synergistes in the cesarean birth. And so we don't, what does this actually mean? That's hard to tell at this point. I think there's still a lot of research to be done. However, we do see that there are differences in how the initial colonization occurs depending on birth type. We also, this is a pretty complicated graph, so I'm not going to try to get you to understand all of it, but we see that actual feeding of newborns affects their microbiota as well. On the left-hand side, and my picture cut off here, I believe my left-hand side here is my breast milk, but I could be wrong. What's important? It's not important to know the exact differences. What is important is that you can see big differences between these two groups. For example, there's a lot more Escherichia found in this left-hand side than there is in this right-hand side. And then there's a lot more Streptococcus over here than we see over here. And then we see a lot more Clostridium here than we see over here. And then we see things over here that we don't see over here at all, right? So we have Heliobacterium and Clostridioides. Well, I guess there's a little Clostridioides over here as well. So you can actually see these bars a little bit over here. They're kind of like very small, so it's hard to tell what's there. Um, the microbiota changes over time, so it kind of goes in these phases where when we look at diversity of the organisms, we see in that first like 3 months there's not a whole lot of difference in diversity, but there's a little increase in diversity as you get older, and then by the time you're somewhere in the range of 3 to 12 years, you've got even more diversity than you did over here. So we see that our microbiota is constantly changing, and that can depend on multiple things. Um, let's see. And so an example here, which I didn't like, I don't have a slide about it, but your microbiota can change based on whether or not you have pets. It can change based on your diet. Um, it can change based on who you live with. So your roommate is probably impacting your microbiota. Um, and so is the food that you eat. And so it's kind of interesting. So we can have disruptions in our normal microbiota. Um, So when there's some sort of perturbation, then we can actually have an issue where our normal microbiota die. And then when that happens, a pathogen's able to come in. So remember, one of the main things that our normal flora does is they competitively exclude unwanted organisms, right? And so if you take an antibiotic, that potentially kills off your host cell or your normal flora, which gives the ability of an opportunistic infection. So if something comes in and causes disease where it normally wouldn't be able to. Um, so we have a couple of examples of places where you can have a disruption in normal flora. So if you were to take an antibiotic, then that might actually lead to a UTI. And the reason it does that is that you have Candida albicans, which is a yeast that lives in the vaginal canal. It's usually kept in pretty low abundance. It's usually kept in the yeast form. It doesn't transition into its hyphal form. But your normal vaginal microbiota, which are these lactococcus and lactobacilli and stuff like that, they're actually able to keep the Candida albicans in check. However, when the normal flora die from taking that antibiotic, then you get this decrease in vaginal microbiota, both in diversity and in biomass, leading to a yeast infection. Similarly, you can get a gut microbiome infection the same way, like C. diff or something like that. So there's this concept of transient microbiota. We don't really consider these part of our normal microbiota. They're just, in and out, right? So they're not sticking around. You can get these from just incidental contact, either, uh, touching somebody or something, or with stuff that you put in your mouth and eat, and they come with their own microbes. Um, these organisms are usually removed just through normal hygiene, so washing your hands and that sort of thing. There are a couple examples of how host microbe interactions influence our own evolution. One of which is malaria, which is a protozoan and or is caused by a protozoan that lives inside of the Anopheles mosquito. It kills like 600,000 people a year. I think it's, the mosquito, because of this, is considered the most um, dangerous animal on the planet. And people who carry the gene for sickle cell are actually protected against malaria. Now, if you have sickle cell anemia, that is obviously detrimental, and it causes significant problems with your ability to carry oxygen in your blood. However, if you're a carrier, you get this slight change in your hemoglobin that makes it less likely for you to get malaria. So that's interesting. And obviously, um, this sickle cell disease is more, or sickle cell anemia carriers and sickle cell disease are more common in areas where malaria is endemic, right? Now, this bit is kind of like add-on. It's in the chapter, and so we'll talk a little bit maybe more about this later, but it's important to know about biofilms because most microbes actually exist in a biofilm form, all right? So a free-floating bacteria would be considered planktonic, right? So it's not associated with anything. However, you can get biofilms, which form on surfaces, and they can be made up of single organisms or a variety of organisms. And so the first step in biofilm formation is attachment. So those planktonic cells will make these adhesion factors, and that'll help them, attach to some surface. Now, this surface could be a catheter. It could be a replacement heart valve. It could be that little pink ring around the bottom of your sink. It could be inside of your, water pipes. The surface could be anywhere, really. And then, of course, one of the most common ones that you deal with is on your teeth, right? You get dental plaque is going to be a type of biofilm. So once it's established or attached, you get growth. Now, this is going to come from the creation of this polysaccharide matrix. And in that matrix, that's going to be like a goo that surrounds the cells, and the cells are going to be able to grow in there and replicate. Now, again, this may start with a single organism, or it may start with multiple organisms. So you can have uh, multiple species in one biofilm. Now you get the uh, development of layers, right? So as you see over here, you get these cells at the bottom near the surface, and then you get more and more layers. And as the biofilm grows, you're going to have like several layers of bacteria to get through, or fungi or whatever in your biofilm. And so the further away from the surface of the biofilm you are, the more protected you are. Now you can get detachment. So occasionally, the microbes in the film are going to be released. They're going to go back into planktonic form. They're going to detach, and they're going to find potentially another surface to colonize, right? And so this is a normal, like part of the life cycle of a biofilm. And here we can see a biofilm, which is on dental floss. This is from a scanning electron micrograph, and that purple stuff is biofilm. So that's just a collection of cells and polysaccharide that stick to your floss. And so when you brush your teeth and you floss your teeth, you're actually rubbing off, you're mechanically removing the biofilm. So you can get biofilm on pretty much any surface, as long as there is something there for the microbes to metabolize. And they are incredibly uh, common in infections. So the NIH, the National Institutes of Health, estimate about 60 to 80% of infectious diseases are from biofilm created in humans. And if you have a biofilm inside your body on like a replacement joint or on a replacement heart valve, they can be very difficult to manage. Um, the immune system can't get into that polysaccharide layer. They can't get all the way through that the biofilm itself. So the stuff near the attachment site are more protected. And same thing for antibiotics. So biofilms are very persistent. All right, and I forgot to add in our objectives as the last slide, so I'm going to go ahead all the way back to the beginning. And so at this point, you should be able to compare and contrast parasitism, mutualism, and commensalism, define the term normal microbiota, and discuss its establishment and roles, and describe how a biofilm forms, discuss the healthcare implication of biofilms. So there should be an objective check, and I look forward to seeing you all in class. Have a good day.